Hello, everyone. My name is April Meadows,
Hi everyone, my name is Akash Kaul.
and this is the summary of our project, Where Are the Filaments Around the Milky Way? This was supervised by Dr. Mark Nyrink, and what we'll be talking about is where these filaments are around the galaxy that we are a part of. And we'll talk about how these slime mold can actually help us find them. To start out, though, what exactly are filaments? These are structures of matter that exist in between our galaxies. They're made of clumps of matter that actually build upon each other to make this connection. Overall, these structures make up something known as the cosmic web. I like to think of it as little nerd candies. For those of you that haven't had it, it's just like a little tiny sugar that's flavored. Very delicious. I highly recommend trying. And these nerds can actually build upon each other into nerd clusters. And then those clusters build to be a giant nerd rope. And this nerd rope is like the filament that we are investigating. There'd be one hypothetical nerd rope in between each galaxy. Now, where we investigated was the local sheet, which is the collection of galaxies that are nearest to us. We're in the Milky Way in the center of this image, and there's a total of 14 galaxies that are of a similar mass in this area. They're about 10 to 16 light years away from us on average. Now, each of these galaxies is actually composed in a relatively flat plane, so that's why we call it the sheet. These are all flat, like a sheet of paper. And they can be visualized in this way, where every pink dot that you see is actually a galaxy. And what we're trying to find is where the filaments, the connections between these pink dots are. And we used the help of an unlikely friend, which Akash will now introduce.
Yes, so to actually help out our analysis, we leveraged these biological organisms called slime molds. And the name slime mold is a bit of a misnomer because it's not a mold and it's not really a slime, but it is quite slimy. They are single-celled protists. And so they're actually just a single cell, but they grow into these vast networks of connected veins, as you can see in some of the pictures here. There are many different species of slime mold, but the one that's commonly used for research is this species called Physarum polycephalum. And there are a few canonical examples of slime molds used in research. A lot of them are related to finding, the shortest path in a maze. But probably the most famous example and probably the coolest one as well is scientists in Japan basically plotted out the Tokyo subway and just let the slime mold sort of run free in that space. And the slime mold ended up mapping out a structure that was very, very similar to what humans had come up with. So, without a brain, without critical thinking skills, they sort of have these network algorithms built into their biology, which is very cool.
Mhm.
And so there's still the question of how does slime mold help for our specific use case of detecting filaments. As I had mentioned earlier, the slime molds have these veinous filament structures that they naturally form, and this sort of directly maps to how we view the cosmic web in its structure. And then there are also some research papers out there that actually use computational algorithms for studying the cosmic web, and those algorithms themselves are based off of slime mold biological behavior. And so all of this basically just informs us that, we think that this is a valuable sort of research project to explore the filaments from the perspective of slime molds.
All right, so how do we do this? Well, taking that picture of those pink dots where the galaxies are from earlier, we actually place oat flakes at the location of each galaxy. And that is the slime mold's food source, which is why we do that. Then we place a slime mold starter right where our Milky Way is, and we let it get to work. Now, to take pictures, we actually had to do that at regular intervals. And so we did a very sophisticated shoebox transportation method where we just put the slime mold in a shoebox and took it to work with us to take pictures and then took it back home. Now, I do have a time lapse of what the slime mold is doing, and it is a bit flashy. So if you're sensitive to that, you might want to look away right now. But here it is. You can see the slime mold starting from the center and then going and finding each one of those oats. Now, Akash can talk about some additional things that we thought about as we were doing this.
Yes. So one of the important considerations for an experiment like this is the actual controls and the experiment setup itself. Right. So for slime molds, they are biological organisms and they are susceptible to fluctuations in temperature and humidity, even in light. And all of those things can affect the growth and ultimately the structures that we see in the petri dish at the end. So because of that, we had to be intentional with how we control for those things. And we also wanted to be intentional with how we didn't control those things, meaning
Mhm.
like we wanted some variability just so we can speak to the structures themselves rather than any specific condition or any specific slime mold growing condition, right? Because fundamentally, this is we're using biology, but it's not a true biology experiment. And so we wanted some variability to be able to speak to things larger than just, the slime mold in a specific condition.
Mhm. All right. So now that we have an idea of how we're doing this, how do we actually analyze it? So here we have our collection of galaxies again, and every single galaxy could be analyzed, or every single picture could be analyzed as a network, and that is done with the slime mold. Every oat would represent a node, and every vein connection would represent an edge. Now, some galaxies are close enough together that they can be represented by one oat, and so that's what the black circles here are showing, is where a single oat would have been placed. Then, once we let the slime mold get to work, it creates this nice little network, which we can then analyze. We once again have our nodes highlighted, and then our connections emerging from that. From there, we can actually make a graph out of it to analyze it mathematically. And we would compare each plate to determine how often these connections were happening. So, for example, between 1 and 2 on this image, how often does that connect? And this was done visually, so it would be nice to actually build upon this with some computational analysis, which Akash will discuss next.
Yes. So in addition to visual verification of the structures, we also wanted to be a little bit more mathematically rigorous when it came to actually analyzing these structures. And so we leveraged many, many different techniques, image analysis techniques using Python and OpenCV. A lot of these were based off of research that's currently out there that has done similar work with slime molds. But fundamentally, we're just trying to process these images into a usable format, right? And so we want to get rid of all the noise. We want to get rid of the background. We want to get rid of things that aren't directly in the petri dish, and really just isolate the slime mold in those networks. And that way, we can really dive into, how are the slime molds? Where are the connections placed, and how do they sort of surface?
All right. So, how often did galaxies actually have connections? Well, these 5 pairs here, which are highlighted on the screen, always connected. We analyzed a total of 9 plates for this experiment because the other plates that we did, they either died too quickly or they didn't have substantial connections for us to analyze. So out of those 9, these 5 pairs always connected. And then from there, 5 pairs actually connected most of the time. So that was considered 7 times or 8 times out of the total 9. And then we had 4 pairs connect some of the times. So 4 times to 6 times out of those total 9. From there, 6 pairs rarely connected. So 1 time or up to 3 times. And out of the all possible connections that could have happened, 15 of those never connected out of the 9 that we analyzed.
Amazing. So again, in addition to visual confirmation and visual analysis, we also wanted to speak to some of these things more mathematically with image analysis. And so on this page, the left image is actually an overlay of the isolated networks from various different images. You can think of this as sort of like an aggregate level view of what these filaments in the cosmic web might look like, within our local sheet. Which I personally, this is one of my favorite images that we generated. And then on the right side, we have um, just a basic sort of network diagram. You can see that the different galaxies within the local sheet are labeled in the nodes. I know that this image is a little bit small, so apologies for that. Um, but from a high level, you can see that there are various connections, right, throughout the local sheet. It's highly, highly connected. And the colors here represent the strength of the connections, right? So we can see a lot of green sort of in the center, and then a lot of sort of yellow and more red around the outside. And this sort of makes natural sense based off of our experiment conditions and other things, which we'll talk about in the next slide. But I think this image does give like a really good high level overview of sort of like how all of these individual galaxies are connected and how they're not connected sort of just like uniformly, but there's sort of branching connections and connections, across the plate and things like that. So yeah, so as I mentioned earlier, there are sort of aspects of the experiment that we can change and that we can improve upon. One thing I had mentioned earlier is standardizing the experimental setup, things like light and temperature and humidity we already talked about, but even to the point of standardizing, the angle that we take the image at, or like the glare in the image, or things like that. Those would all serve to, really benefit us when we do that, excuse me, the image analysis. A second piece here is to help build confidence in the filaments that we've actually identified. This is kind of an interesting point because there's not really an agreed upon like ground truth for the filaments or for the cosmic web. And so there are some interesting questions here about like how do we actually verify the networks that we
Yeah.
discovered, and how do we like know if they're true networks or if they're just sort of noise from the slime molds.
Mhm.
And then yeah, the last piece here is just adding variation. This is what I was alluding to earlier with the fact that most of the like densest connections were in the center of the petri dish. This might just be due to the fact that we would plate the slime mold itself like in the center of the petri dish, and it would sort of branch out from the center. And so there are questions here about like, if we plate the slime mold on the edges, if we plate it in multiple different spots, how would that affect the outcome? And so yeah, so a lot of potential future work here.
Yeah, so to summarize, these filaments are matter that exist in between galaxies and connect the two, and slime molds like polyciferum can actually help us find these filaments. And filaments between certain galaxies are more likely and more often occurring than others, as we saw in our analysis section. And computational analysis can actually show us more about how these connections, like how their strength is, and that kind of stuff. But yeah, thank you all so much for coming to our presentation.
Awesome. Great job to both of you. That's really fun and incredible. Do we have any questions from those who are tuning in? You can raise your hand or type it in the chat. Sanjoy has a question.
April, Akash, phenomenal presentation. Thank you very much. I still find it fascinating that slime mold can give insight. Something as small as slime mold can give insight to something as big as the universe.
Yeah.
Do you think the pattern of connection would change if you just increase the size of the petri dish? Add
Ooh,
your circles further from each other, just like make it 2 or 3 times the size, and that would change things?
probably. I don't know unless we actually try it, and that's like something that we would love to explore in the future is actually accounting for the distance between the galaxies and making that consistent, and also accounting for maybe increasing the number of oats at each galaxy to increase the food source. But yeah, also increasing the petri dish as well to see how long it takes those, like the slime mold to get there and how the network would change in that case. I don't think we could get it on the same scale as the galaxies themselves,
Oh, come on.
but that maybe in the far future.
Awesome. Love it.
Yeah, great work to both of you.